अस्सलाम वालेकुम गाइस। अस्सलाम वालेकुम। आर यू ज्वाइनिंग मी? हेलो हेलो हेलो, क्या ज्वाइन कर रहे हो? स्लो। हेलो। 7:30 का टाइम दिया था भाइयों, जल्द से जल्द ज्वाइन कर लीजिए क्योंकि हम इस वीडियो में बहुत कुछ सॉल्व करना है, इंपॉर्टेंट क्वेश्चंस को सॉल्व करना है। फुल प्रिपरेशन से आया हूँ, आप भी मेरे को सपोर्ट कीजिए जल्द से जल्द। कम फास्ट। Yes, fast. Please, anyone message the video is started in left join pharmacy group. Yes, anyone message please. कोई तो मैसेज करे. Hi, 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 Mayur, hi man. Are you getting my voice? क्या आपको मेरी आवाज़ सुनाई दे रही है? हेलो. Yes, thank you, thank you. Please, anyone text in the Let's Join Pharmacy group. The sir has started the classes, so please come and join. Please, anyone message man. हाँ. मैसेज करेगा. This class really it is very helpful for you. In this class you can ask your doubts. आप अपने डाउट्स पूछ सकते हो दोस्तों. Okay. If you want the doubt for any question in Kannada means inshallah I will explain that also. In Kannada also I will explain. You don't worry about that. Clear? Getting? हाँ. Yes, 8 members are joined. Accepted 20 members. 20 लोग ज्वाइन होंगे तो हम क्लास स्टार्ट कर देंगे एट 7:30 एट शार्प. जितने भी लोग हो हम मेरे को समझ नहीं. एट 7:30 आई विल स्टार्ट द क्लास माय डियर ब्रदर्स एंड सिस्टर्स. हाँ. दिस वीडियो कंटेन्स द answers for the question in the description. After the video you can download that. Okay, here I will show you the answers. I will show you the answers for the particular question, and also I will explain that answer. How can you write easily in the examination? Everything I am going to explain you. Don't worry about that. Okay, 110% my dear young stars, if you observe this video until last thoroughly with your 100% concentration means definitely my dear brothers and sisters, no need to read for the examination. It's enough. It's enough. So give your best here. Invest your only 1 hour for me. Inshallah you can thrash your exam board. Clear? Getting what I am telling? Are you hearing my voice, brothers, sisters? Are you hearing? You can text me in the comment section if you want any doubt means you can text me. You can text me. Paper not visible. Now it is. Now tell me, is it visible? Now is it visible? Hello? Now? Is it visible, guys? Yeah. What's the time now? 7:25. Yes, join friends, join fast. जल्द से जल्द ज्वाइन करिए दोस्तों, जल्द से जल्द हमको अभी वीडियो स्टार्ट करना है. ज्वाइन कीजिए आप जल्दी जल्दी. आपके दोस्तों को बोलिए, आपके फ्रेंड सर्कल में शेयर कीजिए इसको. या आई एम गेटिंग योर टेक्स्ट मैसेजेस. ओके सर, यस सर, थैंक यू फॉर मैसेजिंग, थैंक यू फॉर कमेंटिंग. Really, this comment, what you are commenting, no, it's give me motivation. It's give me motivation. Your comment gives me motivation. It makes me interest to make the class. 5 minutes are remaining. After 5 minutes, I will start my class. Thank you, Mayur. Yes, Shreyas Yan. Brother, you text, man. Yes sir, audible and visible. Hello. Just I will set my mic. Now. Now is it visible? Yes. You don't worry about the question paper. You do one thing. If you are having the 2019 question paper, means you keep that question paper. June 2019 microbiology question paper. You keep aside. You keep on your right side or left side. Yes, I will upload, brother. You don't worry. I will upload the whole what video I am doing. No, जो कुछ भी मैं कर रहा हूँ ना, इसको मैं अपलोड करूँगा. Yes. I will upload, man. I will upload. Really, I will upload in the description. You can go and directly download from that. Okay, and take the pen and paper. It's better for you. Now, let's start. Sir, not the question papers. No, man, it's very important. Why? Because I am telling to solve the question papers means not only in microbiology, in pharmacy, every year the question papers are set on the basis of the previous year. Yeah, for that reason I am solving these question papers. Why? Means only 2 months are left here. Only 2 months. Maybe your examination will be held in the month of May, March, April, May. Okay, in 1.5 month, inshallah in 1 month I will clear all the 4 subjects. After that we will come through the second semester. We will practice those. No, I will provide the question with answers. No, I will provide that. You can read. I will explain in detail in this video. In detail, the particular question is explained in the Hindi language. If you want in Kannada, means I will explain the same word in the Kannada language. Okay. In all the direction, I will support you, my dear brothers and sisters. You don't worry about that. Important questions already are uploaded, brother. You can go and read that. Okay. Personal number. Okay. Let's start the video. Okay. Thank you. Let's start. Yes. Assalamu alaikum, my dear brothers and sisters. Welcome you all to my YouTube channel, Let's Join Pharmacy. See here, today I am here in front of you to discuss about the June 2019 Pharmaceutical Microbiology question paper 2019. Here, first we will discuss about the long essays. Okay, long essays means the 10 marks question. First question is what is pure culture? Write in detail about isolation of pure culture. My dear young stars, this question what you are reading, no? What is pure culture? Write in detail about the isolation of a pure culture. This question is repeatedly asked in our examination. In the year 2018, this question was asked, 2019, 2020, except in the year 2021. Maybe this question will be asked in the year 2022. So be ready with this question. I will tell you. You got the question, no? We have to write what is this pure culture. Write in detail about the separation, isolation of pure culture. Let's discuss the detailed answer with explanation for this. Yeah. Yes, see. See here, isolation and preservation of a pure culture. Unit 1 it is. It comes under the unit 1. First, we will write methods of obtaining pure culture of organism. See here, pure culture contains only one kind of microorganisms. You know what is pure? Pure means is 100% clear, without any contamination, without any addition microorganisms. Means here we are growing only one type of the microorganism. So the name itself it says pure culture. Yeah, you can write the 2 points here. It involves a separation of individual microorganism from mixed population. Importance of this pure culture is it helps for the separation of the individual microorganism from the mixed population. Which one? This pure culture. These are the importance. Now directly we will discuss about the methods of obtaining pure culture. See here, my dear brothers and sisters, basically here 5 methods are there. 5 methods for obtaining pure culture, but for our syllabus only 3 are very important. One is the streak plate method, pour plate method, spread plate method. These are 3 are very important, but you should write this roll tube method, micro manipulator method also. You first write these 5. Okay, streak plate method, pour plate method, spread plate method, roll ट्यूब मेथड, माइक्रो मैनिपुलेटर मेथड, ये पांचों आप लिखिए दोस्तों. समझ में आ रहा है? आर यू गेटिंग माई पॉइंट्स? हाँ. नाउ द फर्स्ट वी विल स्टार्ट विद द streak plate method. See here, in the name itself, streak means what? Streak means a striking. Here what we are doing means we are striking on the culture media. What we are doing? We are striking on the culture media. This type of method is known as a strike plate method, where we are striking culture media on the culture plate. So this method is known as a streak plate method. Simple. See here, don't make your answers complicated, because while your answers are supervising, means checking, no, at that time the supervisor will check your knowledge on the answer paper. He won't see which word you used, which weightageable word you used, because sometimes if you use a different word means not clearly visible. Okay, not clearly visible. Now can you tell me, is it clearly visible? Yes. Dear brothers and sisters, so please cooperate with me. See here, streak plate method you got, no? What we are doing means just we are striking here. Striking, okay, with the help of the needle. Let's start the theory. A small amount of mixed culture is placed on the top of the inoculum needle, which is struck across the surface of the agar medium. Understand this point. Understand the point, very important. Okay, you can write in your own words. This is the agar plate. What is this? For an example, I am telling. This is the agar plate. Now here what you are doing, a small amount of mixed culture is placed on the tip of the inoculum loop. This is the inoculum loop. It is a mixed culture media. What you are doing, just you are inoculating the, which one, you are taking the loop full or the needle full of the microorganisms that should be streaked across the surface of the agar medium. Okay, we have taken the sample, then what we are doing, we are streaking where, on this is the surface, we are streaking on the surface. This is the concept. Then it thins out the bacteria. Some individual bacterial cells are separated and will spaced from each other. Here what if we are start striking with the help of the inoculum means, at that time what happening, my dear brothers and sisters, means the bacterial cell wall starts thickening. The cell wall starts thin. Okay, it degrades, the cell wall thins. Why? Means we are striking. No, we are striking, we are making the full bacteria into half. Why? Because to get clearly morphology, for that we are doing that one. Got it? It is a not most commonly used method, and this strike plate method, it is not commonly used method. Okay, it's not commonly used everywhere. We are not using this method. It is old method, my dear brothers and sisters. It is old method. Modern methods are pour plate method. It is the modern method. Just the three examples I have given. This is the zigzag fashion. Okay, this cut zigzag fashion. This one is of the square matrix type of the streaking method. Different patterns are there. Here again, these, which one, these the two most common methods of streaking are, in this streak plate method, again they are classified as a continuous streaking method. The second one is a discontinuous streaking method. Please put these notes. Yes, I will put. What's your name? Naveen Surpur. Brother, I will put, man. Don't worry about that. Don't worry about the notes. Okay, you don't worry. You just understand the concept. It will helpful for you. I will put the notes. You don't worry. Which one? Continuous streaking method. Here, continuous in the name itself, it is indicating, my dear young stars. Continuous means what? Continuity in the streaking. No discontinuation or the no gap. It's known as a continuous streaking method. Let's study. This method of streaking is continuous in the sense that there is no overlapping of streaking zones. अगर हम एक बार स्टार्टिंग टिप से स्टार्ट करेंगे ना स्ट्राइक करने का, देन वी विल स्टॉप एट द एंडिंग. एक बार स्टार्ट हो गया तो खत्म होगा लास्ट में ही, अंत में ही खत्म होगा. दैट इज नोन एज कंटिन्यूअस स्ट्राइकिंग मेथड. एंड अ लूप इज स्टेरलाइज्ड ओनली वंस इन द बिगिनिंग एंड एट द एंड. ये जो इनोकुलम लूप है ना, नीडल है ना, दिस शुड बी स्टेरलाइज्ड ओनली व्हेन एट द बिगिनिंग. ओके, स्ट्राइक करने से पहले और स्ट्राइक करने के बाद, लास्ट एट अ लास्ट जब हम स्ट्राइक करते हैं ना, उसके बाद में हमको ये स्टेरलाइज करना है. स्टेरलाइज बाय मीन्स ऑफ द हॉट बर्निंग फ्यूम, फ्लेम. ओके, फ्लेम से स्टेरलाइज कर सकते हैं या अल्कोहल भी यूज़ कर सकते हैं. दिस मेथड इज यूजुअली फॉर ब्रॉथ कल्चर माइक्रो ऑर्गेनिजम्स. ब्रॉथ कल्चर मीन्स लिक्विड कल्चर माइक्रो ऑर्गेनिजम्स को हम इस कंटिन्यूअस स्ट्रेकिंग मेथड को इस्तेमाल करते हैं. वी विल यूज हियर विच वन, ब्रॉथ कल्चर. बेसिकली ब्रॉथ कल्चर वी आर ड्राइंग दैट. आफ्टर ड्राइंग व्हाट हैपेंस, यू गॉट द सॉलिड मीडिया, striking what that broth culture. Okay, got this point? This is continuous type. Now discontinuous means what we are doing means we are breaking our continuity here. In this method of striking is a discontinuous and the loop is sterilized between the striking. half आधा कर लिया, फिर लूप को स्टेरलाइज कर लिया, फिर स्टार्ट स्ट्राइक करने के लिए. अब कैसा बोलो, समझ में आ रहा है ना आपको? फर्स्ट लूप स्टेरलाइज करने का है, स्ट्राइक करने का है, बीच में स्टॉप, फिर लूप को स्टेरलाइज करो, फिर स्ट्राइक करो, स्टेरलाइज करो, स्ट्राइक करो, स्टेरलाइज करो, स्ट्राइक करो. हाय सर, प्लीज नोट्स अपलोड ಮಾಡಿ. लेट जॉइन फार्मेसी. आई विल अपलोड मैन, यू डोंट वरी. अपलोड, I will upload it. Principle for strike plate method, it is very important. Okay, see, in this method striking is a discontinuous and the loop is sterilized between striking. This is continuous and discontinuous type. Got it? Now principle for a strike plate method. What is the principle for this? General principle is there. This method is based on the principle of the striking. Yes, in the name itself indicating striking, it is based on that. Then the technique essential essentially dilutes the number of organisms and reduce their density. Here, while we are striking, what happens? The density means the cell wall of those microorganisms degrades, reduces. Sir, new lecture or student? Sorry. Now it's not time to discuss. I will discuss afterwards that. First, give your concentrate here. Okay, first give your concentrate and reduce their density. It allows microbiologists to distinguish and isolate individual bacterial colonies. Basically, you leave it. You just remember the method. Mainly, it's used on the principle of the streaking. Sir, please put the June 2019 paper also. Okay. Here what happens? The method is based on the principle of the streaking. Here what happens? While you are streaking, no, the microorganisms size get decreases. The cell wall get decreases. Okay, so that you can clearly observe in under the microscope, is the pure microorganism is growing there or not. A colony is visible cluster of bacteria. You will get their colony after streaking. No, you will get the colony here. The individual type of the bacteria is grown. All the bacteria originate into the same colony. When each colony के अंदर only one colony में सभी बैक्टीरिया ग्रो करते हैं. इंडिविजुअल कॉलोनीज आर प्योर कॉलोनीज. जो कॉलोनीज इंडिविजुअल रहते हैं, अभी फॉर एग्जांपल इधर एक कॉलोनी, इधर एक कॉलोनी, इधर एक कॉलोनी, इनको हम क्या करेंगे? प्योर सेपरेट कर देंगे. इफ यू सेपरेटेड मीन्स व्हाट हैपेंस? वी गॉट द प्योर कल्चर. Okay, the pure colony is transferred to another plate to produce a pure culture of only one type of the bacteria. इसको हम क्या करेंगे? ट्रांसफ़र कर लेंगे. इसको ट्रांसफ़र करके इनको अलग कल्चर मीडिया के अंदर ग्रो करेंगे. इसलिए क्योंकि ये सिर्फ प्योर कंपाउंड्स को ग्रो करे. ये सिर्फ प्योर माइक्रो ऑर्गेनिज्म, एक टाइप का माइक्रो ऑर्गेनिज्म ग्रो करना चाहिए. वही हमारा मकसद है ना, आइसोलेशन ऑफ द प्योर कल्चर. गॉट इट? दिस इज द स्ट्रेकिंग प्लेसमेंट. होप समझ में आ रहा है दोस्तों? समझ में आ रहा है आपको? टॉपिक्स जो मैं बोल रहा हूँ आपको समझ में आ रहा है लगता है. यस. नेक्स्ट, पोर प्लेट मेथड. जल्द से जल्द हमको ख़त्म करना है. ऑलरेडी इट 7:40. पोर प्लेट मेथड. पोर मीन्स व्हाट हियर? जस्ट व्हाट वी आर डूइंग, वी आर pouring. फॉर एन एग्जाम्पल, वी आर pouring द वाटर टू द प्लांट. इट्स नथिंग बट pour. नो, pour मीन्स डालना. pouring the water to the plant. Same, you understand this principle. In this method, successive dilutions of the inoculum are added into the sterile petri plates. ये मेथड के अंदर क्या करते हैं हम? जो डाइल्यूशन रहता है ना इनोकुलम का, उसको हम स्टेराइल पेट्री प्लेट्स के अंदर ऐड करते हैं. आपको मैं बताऊंगा बाद में किस तरह से करके. See, to these plates, melted and cooled agar medium is poured with rotating the plates, which is then allowed to solidify. फर्स्ट हम क्या करते हैं? प्लेट्स लेते हैं. ओके, प्लेट्स. ये प्लेट को क्या करते हैं हम? अगार मीडियम को डालते हैं इसके अंदर. वी विल पोर द अगार मीडियम इनटू दिस प्लेट विथ रोटेटिंग द प्लेट्स. इस प्लेट को रोटेट करते हुए हम क्या करेंगे? वी विल पोर द अगार मीडिया टू दिस प्लेट. देन वी विल अलाउ दिस अगार मीडिया टू सॉलिडिफाई. इसको हम रखेंगे सॉलिड स्टेट को कन्वर्ट होने के लिए. ओके, देन आफ्टर इंक्यूबेशन द प्लेट्स आर एग्जामिन्ड फॉर द प्रेजेंस ऑफ इंडिविजुअल कॉलोनीज ग्रोइंग थ्रू आउट द मीडिया. उसके बाद हम क्या करेंगे दोस्तों? इन्हीं कॉलोनीज को जो हम प्लेट के अंदर जो स्टोर करे हैं ना अभी, विच वन प्लेट्स विच इज़ देन अलाउड टू सॉलिडिफाई. वो सॉलिडिफाई होने के बाद में वी विल स्टडी द कैरेक्टरिस्टिक ग्रोइंग कैरेक्टरिस्टिक्स ऑफ द इंडिविजुअल बैक्टीरिया देयर. द प्योर कॉलोनीज आर सेपरेटेड, ट्रांसफर्ड इनटू ब्रॉड कल्चर एंड थस प्योर कल्चर इज ऑब्टेन. ये थ्योरी है, बेसिक थ्योरी. द प्रिंसिपल, द मेन प्रिंसिपल इज टू डाइल्यूट द इनोकुलम इन अ सक्सेसिव ट्यूब्स कंटेनिंग लिक्विड अगार मीडियम. इनोकुलम जो हम ले रहते हैं ना माइक्रो ऑर्गेनिज्म, उसको ही डाइल्यूट करना है इधर. फॉर एन एग्जाम्पल, आई एम टेकिंग हियर अ माइक्रो ऑर्गेनिज्म ए. दिस ए माइक्रो ऑर्गेनिज्म शुड बी डाइल्यूटेड सक्सेसिवली. सक्सेसिवली मतलब कंटिन्यूसली वी हैव टू डाइल्यूट दिस with the help of the test tubes. So as to permit a true distribution of the bacterial cell within the medium. किस लिए? Because the bacterial cell is present, no, that should be permit or that should be flows throughout the medium. For that reason we have to successive transfer that, continuous we have to transfer that. Okay, the mixed culture of bacteria is diluted directly in tubes containing melted agar medium maintained in the liquid state at the temperature of 42 degree to 45 degree. Okay, the mixed culture of the bacteria that should be diluted directly in tubes कंटेनिंग मेल्टेड अगार, हम क्या करेंगे, जो मिक्स वाला जो ये फॉर एन एग्जांपल ये क्या है, ये मिक्स वाला कल्चर मीडिया है, इसको क्या करेंगे हम टेक्स्ट ट्यूब के अंदर डाइल्यूट करते चले जाएंगे। फॉर एन एग्जांपल वी हैव टेकन द 4 टेक्स्ट ट्यूब, ये मिक्स वाले को टेक्स्ट ट्यूब ए के अंदर डालेंगे, टेक्स्ट ट्यूब ए वाले को टेक्स्ट ट्यूब बी के अंदर डालेंगे, बी वाले को सी के अंदर डालेंगे, सी वाले को डी के अंदर डालेंगे। इस तरह डाइल्यूशन करना है दोस्तों। बाय दिस वे वी आर डाइल्यूटिंग। ओके, बाय दिस वे इस तरह डाइल्यूट करने से क्या होता है दोस्तों, सेल वॉल जो है ना वो सेपरेट होता है। सेल वॉल गेट्स व्हाट, इट गेट सेपरेटेड and it permit the throughout the distribution of the bacterial cell, throughout distribution of the bacterial cell. For an example, आप शक्कर लीजिए, शुगर को क्या करिए, पानी वाटर के अंदर डाल के आप उसको डाइल्यूट कीजिए। डाइल्यूट मतलब ऐसा एक ग्लास से दूसरे ग्लास को, वो ग्लास से फिर एक ग्लास को, उस समय क्या होता है, शुगर डिजॉल्व होने लगता है आपका। डिजॉल्व मतलब थ्रू आउट डिस्ट्रीब्यूशन हो रहा है। ओके, बैक्टीरियल सेल थ्रू आउट डिस्ट्रीब्यूशन हो रहा है। हियर देयर शुगर गेट थ्रू आउट डिस्ट्रीब्यूशन इन द वाटर। सिंपल कॉन्सेप्ट। इसको हम कितने डिग्री के अंदर रखना है, 42 to 45 डिग्री सेल्सियस। द बैक्टीरिया एंड मेल्टेड अगार आर मिक्सड वेल। ये जो बैक्टीरिया है ना और मेल्टेड अगार को, दोनों को हमें मिक्स वेल करना है, मिक्स कंप्लीटली मिक्स करना है। ओके, द कंटेंट्स ऑफ द ईच ट्यूब आर पोर्ड इनटू सेपरेट पेट्री प्लेट्स अलाउड टू सॉलिडिफाई इनटू द इनक्यूबेटेड। फॉर एन एग्जाम्पल 5 ट्यूब हमने ले आया था ना, किसने ट्यूब, वी हैव टुक द 5 ट्यूब्स हमने लिया है। दैट 5 ट्यूब्स जो है ना, दैट 5 ट्यूब शुड बी पोर्ड इनटू द सेपरेट पेट्री प्लेट्स दैट शुड बी इंक्यूब्यूट। हम उनको इंक्यूबेशन के अंदर रखना है 48 आवर्स तक। उसके बाद उसके अंदर हम कॉलोनीज ग्रो हो रहे या ना हो रहे, किस तरह के कॉलोनीज ग्रो हो रहे, सिंगल टाइप, इंडिविजुअल टाइप ग्रो हो रहे या मिक्सड टाइप ग्रो हो रहे। अगर मिक्स टाइप ग्रो हो रहे तो आवर थ्योरी फेल्स। इफ ग्रोइंग ऑफ द बैक्टीरिया इज ओनली इंडिविजुअल मीन्स देन वी अचीव्ड आवर प्योर कल्चर। समझ में आ रहा ना, दिस सिंपल कॉन्सेप्ट। When the bacterial colonies develop, the isolated colonies develop both within the agar medium and on the medium. Okay, these isolated colonies can be picked up and streaked into another agar plate to ensure purity of the stain. इसको हम दूसरे प्लेट के अंदर डाल के what we will do, we will study the purity of that. We will study the individual cell wall or morphology of the particular microorganism. Okay, let's see the procedure. प्रिपेयर लिक्विड अगार ट्यूब्स, लिक्विड अगार ट्यूब्स को हम प्रिपेयर करना है 4 to 5, देन अलाउ टू कूल एट 45 डिग्री। ओके, ऑल प्रिपेयर वाटर ब्लैंक्स नंबरिंग 1 to 5, 1 से 5 तक हमें 1 2 3 4 5 टेक्स्ट ट्यूब को प्रिपेयर करना है। देन ट्रांसफर 1 ml ऑफ सस्पेंशन ऑफ द मिक्सड कल्चर ऑफ ऑर्गेनिज्म टू वाटर ब्लैंक ट्यूब शेक वेल। ये जो मिक्सड कल्चर ऑफ ऑर्गेनिज्म है ना, हाँ, मिक्सड कल्चर ऑफ ऑर्गेनिज्म वी हैव टू ऐड टू 1 2 3 4 5 टेक्स्ट ट्यूब्स है, 5 टेक्स्ट ट्यूब्स है मिक्स्ड कल्चर ऑफ द ऑर्गेनिज्म। देन शेक वेल, 5 टेक्स्ट ट्यूब के अंदर 1 ml ये मिक्सड क्या है, मिक्सड सस्पेंशन, मिक्सड ऑर्गेनिज्म है ना, 5 टेक्स्ट ट्यूब के अंदर हमें 1 ml 1 ml वी हैव टू ट्रांसफर 1 ml सस्पेंशन फ्रॉम द ट्यूब 1 to ट्यूब 2 एंड रिपीट द सेम प्रोसेस अप टू 5। जैसे कि मैंने बताया था, एक टेस्ट ट्यूब से दूसरे, दूसरे से तीसरे, तीसरे से चौथे, चौथे से 5वें। दिस टाइप वी हैव टू डू। ओके, I will put it in the description, but not now. After the video I will put it. Don't worry about that. Okay, then pour the liquefied agar media in tube to each petri plates containing 1 ml of the diluted suspension. Okay, then rotate the petri plate gently for the uniform distribution of the bacterial cell and allow the medium to solidify. Incubate the plates for 28 to 48 hours. बेसिकली कुछ भी नहीं करना है दोस्तों। ये क्या है, 5 तरह के 5 ट्यूब्स ऑफ टेक्स्ट ट्यूब वी हैव टू टेक। दैट टेक्स्ट ट्यूब विल फिल्ड विद द 1 ml ऑफ द विच वन, 5 टेक्स्ट ट्यूब दैट फिल्ड विद द 1 ml ऑफ द मिक्सड कल्चर। उसी को हम क्या करना है, एक टेस्ट ट्यूब को 2 सेकेंड वन शुड बी ट्रांसफर टू थर्ड वन शुड बी ट्रांसफर टू फोर्थ वन शुड बी ट्रांसफर टू 5th। Okay, the particular test tube should be poured into the into the agar medium. Separate petri plates के अंदर हमको वो टेक्स्ट ट्यूब को पोर करना है। That should be keep for the inoculation for 48 hours. Afterwards, we have to check the growth of the colonies, individual colonies. This is the pour plate. Simple method. The next one is the spread plate method. See, very important, this question has been asked for 5 marks also. Write in detail about the spread plate method. Okay, we are very close to our first answer. In this method, microorganisms are spread over the solidified agar medium within a sterile and L-shaped glass rod, that is the spreader. Just here what we are doing means, with the help of the spreader, we are spreading the microorganisms where over the agar medium with the help of which one? This is the spreader. Now let's study the principle of this. The spread plate technique involves using a sterilized spreader within a smooth surface made up of the metal or the glass. Okay, we have to take the spreader that should be sterilized spreader, okay, within a smooth surface. It should be made up of the smooth surface with the metal or with the glass. Then a small amount of bacteria applied in a solution over a plate. A small amount of bacteria should be applied over a plate. A plate needs to dry at the room temperature so that the agar can absorb the bacteria more readily. Simple, simple. कुछ भी नहीं करना, स्प्रेडर लेना है, स्प्रेडर लेके क्या करना है, दिस स्प्रेडर शुड बी प्लेस्ड विद द माइक्रो ऑर्गेनिजम्स and that should be spreaded where? Over the agar petri plate. This is the simple principle. Okay, wait 2 minutes. Yes, now let's study the procedure. Take 3 nutrient plates and a label with the name of the organism to be inoculated. We have to take the 3 nutrient plates. Inoculate the plates within a loophole of the given organism. What we have to do? We have to inoculate those plates with the loophole of the given organisms. Then place plate 1 on the turntable. Means that plate 1 should be revolving. घूमते रहना है उसको. हम क्या करना है? That revolving table should be placed that contains this petri plates. 3. We have to take 3 revolving table. Then sterilize the spreader by putting it first in ethanol, then on the flame of the Bunsen burner, and cool the rod for 30 seconds. First we have to sterilize the spreader by putting the alcohol. After that, first in ethanol, then on the flame. देखिए, it's very important. Remember here, after putting the ethanol, just don't go and directly hit that which one? That spreader, because alcohol, it is the very dangerous. It catches the flame very suddenly, quickly. So for that reason, you stop for 2 to 3 minutes. Then you go for flaming process. After that, cool the rod for 30 seconds. 30 seconds. Okay. Then remove the lid of the plate and spin turn the table. Means you have to remove the lid from the agar plate. Then start the revolving machine and put the spreader like this fashion. Then touch the spreader gently on the surface of the agar. Move it on the forth and back to spread the bacterial source on the agar surface when the turntable is spinning. आपको ऐसे आगे पीछे करते रहना है. Okay. Then when the table stops spinning, put the lid over the half of the petri plates. Sterilize the spreader again and repeat the same process for the 2 plates. जैसे हम 3 प्लेट्स लेते हैं ना, उन्हीं 3 प्लेट्स को इसी तरह का प्रोसीजर हमको अपनाना है. इनका इंक्यूबेट ऑल द 3 प्लेट्स 37 डिग्री फॉर 24 आवर्स. ओके, एक दिन के लिए हम उसको इंक्यूबेट करते हैं, उसको रखते हैं. ओके, क्योंकि डेवलपमेंट देखने के लिए कल्चर करते हैं हम उनको. देन दिस हियर just what we are doing, diluted bacterial suspension, one full inoculated here. Then it should be revolving where? On the revolver. Then with the help of the glass spreader, what we are doing? We are spreading that. We are keeping for 24 hours. After that, we will see the isolated colonies development. Yes, my dear brothers and sisters, this we completed the which one? First answer. Now the next question is, you got it, no? This one. Yes, I will upload the same answer what we have wrote, no? The same what we have studied, no? The same answer in the description. You can go and download from that. Next one is explain the principle, procedure and application of any one thermal method of sterilization. Here I will discuss the hot air oven. Okay, hot air oven. Basically, any one method they asked, but for 10 marks it's not suitable. It's not suitable for 10 marks. So better you write 2 methods of the sterilization, thermal method of sterilization. Let's see that which method, thermal methods. Hot air oven, it is also one of the thermal method. Flaming, clear. Any 2 easy method we can write here. Autoclave, identification of the bacteria. Yes, sterilization. See, basically sterilization, what is this sterilization? Sterilization here, just maintaining the sterility. The process in which we are making our production free from pathogens or pyrogens, free from all type of the microorganisms. Even the progeny should be absent in our preparation. Okay, that process is known as sterilization, the process that removes, kills or deactivates all forms of the life. It may be bacteria, fungi, virus, spores, any time. What we are doing, सबको खत्म कर डालेंगे, सबको खत्म कर डालेंगे. That process is known as sterilization. Okay, this is very important. You observe sterile water in the medical shops. Sterile water is used mainly for the infusion purposes. For what we are using sterile water? For the infusion purposes, because it contains 0% pyrogens, 0% microorganisms, no microorganism development. For that reason, yeah. See here, basically dry heat method, thermal method, dry heat method, then flaming, hot air oven. These all are the thermal methods of the sterilization. Let's study the hot air oven here. See here, it is the most widely used method of sterilization by dry heat method. Which one? That is of the hot air oven. Dry heat method, thermal method, we are supplying the temperature here. Okay, just the diagram is very simple for this. I have not wrote the diagram, but I will upload in the description. See here, simple. This is the door. Okay, these are the shelves, plates. These plates contains the containers or the shelves. Why means to keep the microorganisms. Petri plates are here. Petri plates. Okay, and it is I have better notes. Okay. Petri plates. We are keeping your Petri plates in the shelf. My dear brother, understand the concept. Concept is very important here. Please understand the concept. Yes, it is the most widely used method of sterilization by dry heat. With the dry heat, it is one of the most common method. The substances which are not degraded by heat are set to sterilize at 250 degree by hot air oven. See here, basically what happens, many substances, more than 30 to 40 substances, they get sterilized at 100 degree, 120, 150 degree. But some microorganisms, they are not degraded, they are not reduced their action or their cell wall. Okay, see here now, is it visible? Some microorganisms, they are not sterilized at 100 degree or 150 degree. So at that time we are using 250 degree Celsius by hot air oven, 260 degree by hot air oven. Yes, hot air oven. Just what we are doing, we are supplying the hot air. Oven you observe, no? Oven you observe, no? See here, egg puff. You keep the egg puff in the oven, Domino's pizza in the oven, the same oven we are using here. Okay, but it is modified. Normally at temperature of 160 degree for 2 hours, spore as well as small microorganisms will die. Normally at which temperature? 160 डिग्री टेम्परेचर के अंदर बेसिकली 2 घंटों को हमको रखना है. फॉर द 2 hours if we keep means the small spores, also the small microorganisms will die. Clear? It is ideal method of sterilization of the glassware, surgical equipments, then swab sticks, etc. The sterilization temperature and holding time for sterilization in hot air oven. See here, basically we are using here 3 basic type of temperature. First, for 3 hours at the degree of 140 degree Celsius, we are heating for 3 hours. And for 1 hour, 160 degree Celsius. For 30 minutes, 180 degree Celsius. At last, for half an hour, what we are doing? We are heating up to 180 degree Celsius. 180 degree Celsius we are heating here. 180 degree Celsius. Okay, here what happens? minute pore also, millipore also get degraded here at 180. It is one of the very good method for sterilization, my dear brothers and sisters. Which one? This hot air one. First, what you have to write? You have to write these all, means which are not degraded. It should be degraded at 250 degree by hot air one. Normally we have to use normally the temperature of 160 for 2 hours will kill all the small type of the bacteria. Okay, then here this method is basically used for the sterilization of the swabs, instruments, surgical instruments, glassware. Then here the main points we have to remember: this sterilization basically carries into 3 steps: 3 hours for 180, 1 hour for 3 hours for 140 degree, 1 hour at 160, 30 minutes should be at degree Celsius, 180 degree Celsius. We have to heat that. Clear? Now, sterilization control of hot air one. Generally, 2 methods to control this sterilization, means we can maintain the sterility of the compound or sterility of any equipment by 2 methods. One is chemical control, biological control. Chemical control, what we are doing means Brownion's tubes containing red solution is placed within the oven. Brownion's tube. Okay, Brownion's tube, that should be red solution, containing red solution. We have to place where? In the oven. A green color is produced after 60 minutes at 180 degrees Celsius, which indicates proper sterilization by dry heat. See here, with keeping the instrument which we want to sterilize now, that instrument should be kept inside the hot oven. Okay, side to that we have to keep Brownion's tube, that should contain the red solution. After attaining 160 degrees Celsius, what happens? After 1 hour, the red color changes to green color. This helps to identify the compound is sterilized. Okay, this is the chemical control. How can we understand or how can we know the sterility? But now what happens? The all are modified. The techniques are modified here. Okay, we can easily know the sterilization. नाउ बेसिकली दीज टेक्निक्स, अब जो टेक्निक्स है ना वो सब कुछ बदल चुके हैं अभी, सभी मॉडर्न, ऑल आर टेक्निकली और कंप्यूटराइज्ड. क्लियर? देन बायोलॉजिकल कंट्रोल. An envelope containing a filter paper strip inoculated with 10 spores of Bacillus subtilis is placed within the oven. 10 spores we have to keep where? Inside the oven. ओके हम किसके अंदर रखना है उनको फिल्टर पेपर के अंदर फोल्ड करके हमको वो जो बैसिलस सबटाइलियस को हम हॉट ओवर के अंदर प्लेस करना है. आफ्टर स्टेरिलाइजेशन प्रोसेस द स्ट्रिप इज रिमूव्ड एंड इनोकुलेटेड इनटू ट्रिप्टोन सोया ब्रोथ के अंदर हमको क्या करना है? इनोकुलेट करना है. एंड इनोकुलेटेड एट 37 डिग्री फॉर 5 days अंडर द एरोबिक कंडीशन. ओके, वेरी इंपॉर्टेंट. एरोबिक इज वेरी इंपॉर्टेंट हियर. What we are doing after the completion of this sterilization process, that strip should be removed and that should be inoculated, grown where? Into the tryptone soya broth for 5 days under 37 degrees Celsius in aerobic condition, air के अंदर, in the presence of oxygen. Absence of a Bacillus subtilis indicated the sterilization of dry heat is perfect. Dry heat perfect हो गया बोल के हम बोल सकते हैं. When the Bacillus subtilis is absent there. Clear? This present or not we are using by means of which one? Indicators we are using. Simple indicators or staining procedures we are using for that. Gram staining techniques we are using for this. Okay, you got this point? Yes, this is the simple thermal one. We studied hot Europe. You can physical methods, moist heat method, filtration, radiation, sound and ultrasonic vibration. These all are the methods of sterilization. In this we have selected the thermal method, moist heat sterilization. Okay, with this you can write the autoclave. In 5 marks I will discuss that autoclave, you don't worry. Yes, I think we have completed the 2 questions. 2 equations we have completed, my dear brothers and sisters. How many? 2 questions. That is of a first one, what is pure culture? Write in detail about the isolation of pure culture. Explain the principle, procedure and application of any one thermal method of sterilization. We have completed these 2. Then the next one is explain different methods for evaluation of a bacteriostatic disinfectant. How can we evaluation of bacteriostatic disinfectant? Different methods we are using for disinfection. Different methods we are using, different evacuation methods we are using. We can discuss that. Okay, can we discuss in this video or in next video? Enough for today's? Please text me, my dear brothers and sisters. Is it enough for today or we continue? Hello. These are two questions answers I will provide in the description. You can go and download from that. Okay, you can easily go and download from that. Took 45 minutes class. I think so it's enough for today's class. Inshallah, in tomorrow's class I will discuss these third one question with some of the 5 marks. At last tell me, sir, how represent the paper in examination? See, representing the paper in the examination is very simple. Representing the examination, the paper in the examination is very simple. You remember one point. For an example, they have given the write a note on hot air oven. Basically, you first write the diagram for that hot air oven construction. After that, you write the principle. After principle, my dear brother, after principle, you have to write some of the theory or working procedure. Working means this only. We have to keep which one? The microorganisms where inside the shells. Okay, that should be placed with the Brownian solution, red solution. After some time, we have to continue heating the procedure for 160 degree for 60 minutes. This is all procedure you have to write there. Diagram is very important for all the questions. If you know the diagram means you just describe the diagram, friend. Why means diagrams give better explanation than your writing, than description. So please visible your diagram. आप डायग्राम लिखिए, डायग्राम को ड्रॉ कीजिए. डायग्राम वेरी इंपॉर्टेंट, इट प्लेज अ वाइटल रोल हियर. ओके, yes, I think it's enough. Today we have completed the 2 questions in 10 marks. See here, in tomorrow's class, inshallah, I will discuss the third one, and for 5 marks, I will discuss all the 12 questions in tomorrow's class. Okay, it is a short class because I want to check whether it is suitable for me and for you. Yeah, thank you, man. इसलिए मैंने चेक करने के लिए ये क्लास को किया था दोस्तों. थैंक यू, यू सपोर्ट मी वेरी वेल. अच्छा आपने सपोर्ट किया, कमेंट किया, मुझे मोटिवेट किया दोस्तों. रियली इसी तरह होते रहा तो रियली मुझे भी बहुत पसंद आएगा क्लासेस करने के लिए, आपको सुनने के लिए भी पसंद आएगा. देखिए दोस्तों, अगर आपका डाउट है, इफ़ यू आर हैविंग एनी डाउट मीन्स यू कैन आस्क. इंशाल्लाह आई विल क्लियर योर डाउट्स. Analysis you have said, bro. Yes, madam. Question paper analysis means see with the question paper analysis we will discuss the answers. Why I am discussing the answers means see just to remember why I am discussing the answers with the analysis means many students they don't know the appropriate answers, how to write in the examination. Many students, they know everything about the topic, but they don't know how to describe, how to write in the answer paper particular answer. Okay, for that reason I am came here to tell you, to describe you how to write in your examination. लिखते कैसे है, लिखा कैसे जाता है, उसी के लिए मैं आया हूँ दोस्तों. ओके, आप इस तरह से लिखेंगे तो 110% यू विल पास योर एग्जामिनेशन. प्रैक्टिस इन दिस मैनर. ओके, इफ यू आर हैविंग एनी डाउट मीन्स यू कैन व्हाट्सएप मी टू माई नंबर 7353649264. I will upload the notes in the description. Go and sir. Yes, thank you, man. I can upload the answers in the description. You can directly go and download from that. Then inshallah in tomorrow's class we will discuss with the third question. If possible, we will discuss all the 5 marks questions. My ambition is, or my goal is to complete in 1 month all the 4 subjects with passing strategy for you, with passing marks for you. We will do after the pattern of this question papers, discussing the all the question banks. We will study that. You don't worry about that. Okay, we will study that. Clear? We will study that. And if you give your concentration 100% here means definitely you will score maximum marks in your examination. ओके और एक बात, ये जो मैं वीडियो बना रहा हूँ ना दोस्तों, हाँ, व्हाट आई एम डूइंग दिस वीडियो नो, रियली माय डियर ब्रदर्स एंड सिस्टर्स, इस तरह का वीडियो आपको किधर भी नहीं मिलेगा. लाइव सेशंस डिस्कस करना, आंसर्स आपके साथ डिस्कस करना, ये आसान की बात नहीं है. यू विल नॉट गेट एवरीवेयर दिस. हर जगह ये नहीं मिलता, हर जगह ये मौका नहीं मिलता. समझ में आ रहा है ना? हर जगह ये मौका नहीं मिलता. आपको मौका मिला है, इसको यूटिलाइज कर लीजिए दोस्तों. यू यूटिलाइज दिस अपॉर्चुनिटी. ओके एंड दिस मच यही कहना था आपसे दोस्तों. और कुछ शेयर करना है तो नेक्स्ट वीडियो में इंशाल्लाह हम शेयर करेंगे. ब्रदर आई एम फ्रॉम SCS कॉलेज ऑफ फार्मेसी, SCS कॉलेज ऑफ फार्मेसी, हरपनहल्ली. ओके yes, biochemistry notes you want, inshallah I will do that, you don't worry. After the completion of these, here हम धीरे धीरे चलेंगे दोस्तों. 1 महीने के अंदर मेरा टारगेट है कंप्लीट करने का, इंशाल्लाह मैं कंप्लीट करूँगा. इसके बाद जो है ना 1 मंथ के बाद, हाँ, आफ्टर 1 मंथ जो है ना दोस्तों, वी विल स्टार्ट सेकेंड सेम. इफ़ यू आर इंटरेस्टेड मीन्स यू कैन अटेंड द क्लासेस. जो अगर आपके बैक है तो आप अटेंड कर सकते हैं. इज दिस वीडियो इज़ हेल्पफुल? प्लीज़ गिव मी फीडबैक. हेलो, अच्छा लगा या नहीं लगा? अच्छा लगा. थैंक यू मैम, थैंक यू मैम. Definitely we will complete, sir. Inshallah we will complete. We will complete definitely. With your support, we will complete. Yes, I will upload the video. I will upload the notes in the description. You can download from that. Thank you. Assalamu alaikum.